Welcome to this lecture on the fundamentals of genetics. Today, we are going to explore a truly fascinating topic, the basis of sex determination. Have you ever wondered how nature decides if a developing embryo will become male or female? It is not just random chance. There are highly specific, organized rules that govern this critical decision. By the end of this video, you will understand the genetic and environmental triggers that flip the biological switch toward maleness or femaleness, and why this balance is absolutely essential for the survival of species across the globe. To understand how this works, imagine you are building a house, and the architect's blueprint comes with a special sealed envelope. Depending on what is inside that envelope, the house will either have a blue roof or a pink roof. In biology, this sealed envelope represents the mechanism of sex determination. It is the specific set of instructions that tells a developing embryo whether to grow male or female reproductive organs. For a long time, people thought this was just random chance, much like flipping a coin in the dark. However, nature has very specific, highly organized rules for making this critical decision. Now that we have our blueprint analogy, let us look at how this actually happens in living things. The basis of sex determination refers to the underlying biological mechanisms that dictate sexual development. In most animals, including humans, this is a strict genetic process. It is controlled by specific sex chromosomes, such as the X and Y chromosomes. These genetic packages carry specific genes that act as triggers. Once activated, they set off complex developmental pathways that ultimately lead to the formation of male or female physical traits. This is the system you are likely most familiar with. While genetics play a huge role in humans, this brings us to a completely different mechanism found in nature. In some reptiles and fishes, sex is not determined by genetics at all. Instead, it is determined by environmental factors. For example, the temperature at which an egg is incubated can act as the ultimate deciding factor. If the nest is hot, it might produce one sex, and if it is cold, it might produce the other. Understanding this basis means looking at the exact triggers, whether a specific gene or an environmental cue, that flip the biological switch. Before we dive into the mathematics of these systems, we must ask, why does this concept matter so much in the grand scheme of biology? Without a reliable system for sex determination, species could not maintain a balanced ratio of males to females. This balance is absolutely essential for sexual reproduction and survival. In the real world, understanding these mechanisms allows doctors to diagnose and treat sex chromosome disorders in humans, such as Turner syndrome or Klinefelter syndrome. It also has massive implications in conservation biology. For instance, knowing that sea turtle sex is determined by sand temperature helps conservationists protect them from global warming, which could otherwise cause all hatchlings to be born female. Now that we understand the importance of these mechanisms, let us look at the mathematics behind human sex determination. In humans, the defining genetic factor for maleness is the presence of the Y chromosome. This formula calculates the probability of a child being biologically male. Conceptually, it comes from multiplying the independent probabilities of the gametes, the sperm and the egg. P of M is the total probability we want to find. P of Y is the probability that the father's sperm carries a Y chromosome. P of X is the probability that the mother's egg carries an X chromosome. If the father's probability of passing a Y chromosome increases, the overall chance of having a male child increases. Let us see this formula in action with a concrete example. We are given a human couple expecting a child, and we need to find the mathematical probability that the child will be male. We chose this formula because human sex depends on the combination of gametes. First, the mother can only pass on an X chromosome because she has two of them, so her probability is 1.0. The father has one X and one Y chromosome, meaning he has a 50% chance of passing on the Y chromosome, so his probability is 0.5. We multiply these independent probabilities together, 0.5 times 1.0. The final answer is 0.5. Physically, this means nature flips a perfectly fair coin, resulting in an equal likelihood of having a boy or a girl.
We have just proven that human populations are roughly split evenly due to the Y chromosome. But what about other organisms? While human sex is determined simply by the presence of a Y chromosome, other species use a more mathematical approach. Enter the fruit fly, Drosophila. In these fascinating insects, sex is not just about having a specific chromosome. Instead, it is determined by a delicate mathematical balance between the X chromosomes and the standard non-sex chromosomes, which we call autosomes. This brings us to a famous biological rule known as Bridges' genic balance theory. This mathematical approach leads us directly to the genic balance theory formula. Conceptually, this formula represents the biological tug-of-war between genes on the X chromosome and genes on the autosomes. Let us break down the symbols. S represents the sex index, which ultimately dictates the physical sex of the fly. X represents the total number of X chromosomes present in the fly's cells. A represents the number of sets of autosomes. What happens physically when these variables change? If the value of X increases relative to A, the index goes up, pushing the fly toward becoming female. Conversely, if the value of A increases relative to X, the index goes down, pushing the fly toward becoming male. Now that we have the formula for the sex index, we need to know how to read the results. The calculated value of S acts as a sliding scale for sexual development. According to established genetic rules, if the index is exactly 1.0, the fly will develop as a normal female. If the index is exactly 0.5, the fly will develop as a normal male. But here is where it gets incredibly interesting. If the value falls anywhere strictly between 0.5 and 1.0, the fly will develop as an intersex fly. This means it will have a fascinating mixture of both male and female physical characteristics. Let us apply these rules to a real laboratory scenario. We are given a mutant fruit fly with 2 X chromosomes and 3 sets of autosomes, a rare condition known as triploidy for autosomes. We are asked to determine its biological sex. We use the genic balance formula because Drosophila sex depends on this exact ratio. First, we identify our variables from the problem. X equals 2 and A equals 3. Next, we substitute these values into our formula, dividing 2 by 3. The calculated sex index is approximately 0.67. Because this value falls strictly between 0.5 and 1.0, our final answer is that this specific fruit fly will develop as an intersex fly, proving that sex in Drosophila is indeed a sliding scale. Having calculated the genetic ratios, let us visually compare these two vastly different systems. On the screen, you are looking at a side-by-side comparison of genetic and environmental sex determination. On the left, we have the classic Punnett square for the human XY system. You can clearly see the mother's X chromosomes on top and the father's X and Y on the side, perfectly illustrating that 50-50 split we calculated earlier. Now, look at the graph on the right. This represents temperature-dependent sex determination in turtles. The horizontal axis shows the incubation temperature in degrees Celsius, and the vertical axis shows the percentage of females. Notice the blue curve. At lower temperatures, the hatchlings are predominantly male, but as the temperature increases past 28 degrees, the curve rises sharply, reaching 100% females at higher temperatures. This diagram beautifully teaches us that while genetics use strict inheritance, the environment can act as a powerful, sliding biological switch. We have covered a lot of ground today, so let us reinforce the most important ideas. First, the basis of sex determination is a fundamental biological system that decides whether an organism develops as male or female. Second, we saw that this can be controlled by strict genetic inheritance, like the XY system in humans that guarantees a balanced population. Third, we learned that it can also be a delicate mathematical balance, as proven by the genic balance theory in fruit flies. Finally, we explored how the environment itself, such as temperature, can act as the ultimate deciding factor. Understanding these diverse mechanisms gives us profound insight into how life maintains balance and adapts. You have now mastered the fundamentals of sex determination.